chapter's worth of information because it doesn't, I've tried it myself. I'm never successful. Slow and steady wins the race. So please make sure that you're doing that. You've got all this fun stuff at your disposal that you can or you can choose not to use. I will not be offended. You do what works for you, and that'll be wonderful. That test, by the way, will be a 50-question, multiple-choice Scantron test, as are all of your lecture exams. They're all on Scantron. They're all multiple choice, so you do need to buy Scantrons in order to take those tests, which you can purchase in the bookstore, or you can get them online. If you want to buy them in bulk, you can get them on Amazon for cheaper, but then you probably have to buy like 50 of them, which, I don't know, unless you want to just like make it rain Scantrons in the classroom, it's probably not going to be successful for you. So chapter 7 is the control of microbial growth. So chapter 6, it's all about sweet little baby bacteria. What can we do to make you happy? What pH can we make it? What temperature? What oxygen, right? All of these things. And now we're going to see how knowing all of those things means that we can control the growth of microorganisms. And the reason we say control and not kill is because not everything we do is actually meant to kill microorganisms. Sometimes we're just trying to stop them from growing. So we don't say kill, we say control. So control means in some way affect binary fission. That could be just halting binary fission. That could be making it so that binary fission will never happen again. So we're going to look at different things that affect how easily we can control microbial growth, and then different methods of controlling microbial growth, many of which we will be doing in lab over the next few weeks. So that's exciting because you get to do what we've been talking about. So first of all, some terminology. This is a table from your textbook. Notice I put a big giant red line through one of them because we don't care. Right, so 1, 2, 3, 4, 5 words. If you like to make flashcards, this is kind of going to be your jam because there's lots of terminology in chapter 7. So you can do some fun online flashcards, Quizlet. You can do that, old person flashcard where you actually buy index cards and write things on them. But you will want to commit these 5 terms and these 3 descriptions to memory. So first of all, sterilization, right? So sterilization means you've killed everybody. There's a little caveat there with these things that are called prions that we're going to study in chapter 13. And a prion is a crazy, crazy, crazy thing. It's a protein that causes infection. And it's just a protein. Have you ever heard of mad cow disease? Mad cow disease is caused by prions, right? We're going to talk. I've been researching prion diseases. It's been really exciting, and I'm going to share that with you. I've been sharing it with my children at the dinner table. They've not been excited about that. But, that's what happens. So, sterilization, with the exception of those prions, right? We can just kind of throw that exception away. Sterilization means everything's dead. I'm going to flip all the way down to the bottom because I want to compare that term sterilization to the term sanitization. So a lot of people confuse and think these are equivalent terms, that to sterilize and to sanitize means the same thing. Sanitary means clean. So you're cleaning. You're not sterilizing. You are reducing the microbial load in order to make that area clean. Right, so kind of like pasteurizing cleans food, sanitizing cleans surfaces. So when you spray the surface with Capicide, you're not sterilizing it, you're sanitizing it, you're making it clean. You're using some form of control to reduce the number of microorganisms to a safe level. So a lot of times this is thought about with food microbiology, but sanitation also exists in hospitals, right, which is more immediately of a concern to you because you all want to go into public health, right? So when you get, right, like, let's just be super gross, right? Somebody dies of an infectious disease in a hospital in a gurney, right? They don't sterilize that gurney. They clean it. They sanitize it, right? They wipe all of it down, right? A lot of times they let those disinfectants stay for a long time, which we're going to talk about in a few minutes. They sanitize the room. They may sterilize certain things that that person has come into contact with, but primarily the effort is to sanitize, to clean, right? So to get rid of those things so that they're at a safe level. They're below what we're going to learn about later on is called the infectious dose, right? There's not enough of them to make anybody sick, and that's the goal, right? So that's a goal both in food safety, right? So if you've ever worked in food handling or food prep, or worked in a restaurant, you see these big cool looking dishwasher things, and those sanitize, right? So when you get your fork at the, you're sitting in the restaurant, you get your fork and there's a little weird thing on it, and you can at least say to yourself, well, it's a sanitary thing. So it was someone's food, but at least it's sanitary now, right? So sanitary is not sterile. Then we have a couple of different terms, two more that people actually use interchangeably, and those are disinfection and antisepsis. And if you use them interchangeably, you're wrong. Also, if you think, right, you see this word vegetative, and I don't know why, but students, a lot of times what I'll get when I ask people, what's the difference between disinfectant and antisepsis, they'll say, oh, this one's for vegetative cells and this one's not. Here's the difference. Can you rub it on your body? If you can rub it on your body, it is an antiseptic, which is meant to stop the growth, antisepsis, sepsis means growth, of organisms on you. Disinfection is meant to control growth on some inanimate object. So we're not saying one kills and one doesn't, or one is anything, right? What we're saying is, can you use it on living tissue? Is it safe on skin and mucous membranes? If it is safe on skin and mucous membranes, then it is an antiseptic. There are lots of things that are really good at the destructive of vegetative pathogens, but because you're the bigger wuss in this whole triad of you and microorganisms and chemicals, you usually would die before the microorganisms would, right? So like bleach, great disinfectant. But you can't rub bleach on your skin because it will damage your skin. Cavicide, great disinfectant. A couple of you have had spills. And I don't say spray Cavicide all over yourself. I say go wash your hands with soap and water for 30 seconds, right? Because soap is an antiseptic, Cavicide is a disinfectant. So the difference between the two is, can I spray it on myself? Right? So notice everything about those definitions is the same. Antisepsis adds on living tissue, skin and mucous membrane. So that's the difference. The last one, degerming. Degerming is kind of an interesting term. It's used in a very small area. And because it's used in a small area, the antiseptic that is used may not be safe for widespread use, but might be safe for that small area. So degerming would be to remove microorganisms from a little area, right? So like if you're gonna get a shot, or you're gonna get your blood drawn, they'll take an alcohol wipe or maybe an iodine wipe and they wipe it on your skin, and then they inject into the skin, right? The goal there is to remove more than just transient microorganisms because you're about to breach the skin. So you will sometimes use something that is a little harsher, right? Like alcohol. Alcohol is not really considered an antiseptic. It's considered a disinfectant because it's pretty harsh, right? Like we're gonna do a lab where we do alcohol wipes and alcohol gel and hand washing and kind of see how each of those works. The problem with alcohol is that it's damaging skin and mucous membranes. Now you'd really be able to tell, if you took an alcohol wipe and like rubbed it in your nose, you would definitely be able to tell if this thing is damaging your mucous membrane, right? Don't try this at home. But people rub alcohol on their hands all the time, and what they don't realize, because the outer layers of your skin are dead, is that you're damaging them, right? You're drying them out, you're causing minute cracks in the skin, which will actually make you more likely to contract other microorganisms. So we're going to look at what those data look like, and we want to keep in mind, right, that we don't want to damage skin, because when we go to chapter 16, we're going to find that skin is your best defense. Intact skin, great defense. Using alcohol all the time in your skin dries your skin out, and when you have those minute cracks and you're working with a patient who has like, some nasty thing, then that grows into minute cracks in your skin and can make you sick. So degerming, we sometimes use things that are more harsh than we would use generally, because we're trying to hit some tiny area of the skin for a particular purpose, right? Like an incision for an operation, surgery, right, or shots or something like that. So degermine, small area, right? And then these other three down here, I decided to show you graphically because I want you to get used to seeing something happening over time, because we saw a lot of that in chapter 6. We're going to graph that in our lab 10. So I want you to get used to looking at this, right? So if we look at this one, it's got more labeling, right? It's got cell number over time, and it's got two different measurements, viable cell count and total cell count. Now, if you hadn't been here on Thursday, you might think, who cares? What's the difference? But what do you know now? You know viable, right, are capable of reproducing. Total means that also includes dead cells. And if you've just gotten done disinfecting, do you care about the dead cells? No, because they're not going to make you sick, right? So notice these are two different counts. So the viable cell count could have been spread plate and serial dilutions, right? Because you can serially dilute a culture and then put those dilutions onto plates and count colony-forming units, right? Total cell count could be something like the spectrophotometer. The spectrophotometer, where you're just looking at live and dead cells, right? Or that could be a microscopic count, where you're just looking at the microbes individually, right? Under the microscope, but you can't tell live from dead. So this agent, which is something static, right? That's a prefix. So microbistatic, virustatic, fungistatic, bacteriostatic, static, stasis, right? When someone says, let's just keep the status quo, right? Just keep it like it is. So stasis means to stay the same. Static means that the number of cells over time doesn't change, right? So at this point, you've added some microbistatic, that's sort of our catch-all phrase for microbes being held in stasis, some microbistatic agent, and neither the viable cell count or the total cell count increases over time, right? So when you get your plates out of the incubator, same number of colonies on the plates. When you get stuff out and you put it under the microscope and you look, it's the same number of cells. Nothing changes over time. Because all you've tried to do is stop binary fission. And that could be your goal, right? I mean, even when we take antibiotics, if you're a healthy person and it's a non-life-threatening infection, you really, all you need to do is this. The problem would be if this went unchecked, that would overwhelm even a healthy person's immune system. But if the antibiotic can just slow or stop the reproduction, then your immune system can take care of that, right? And that's really good because sometimes that means that we can take things, right, at lower doses, and that means that things don't end up becoming antibiotic resistant because we end up with shedding antibiotics and things like that, which actually happens with people, which we'll talk a lot more later on. So static agents stop the growth of the pathogen, stop the binary fission. Cidal agents, right, that's probably one that you can make out without even thinking about it, right? Homicide is killing homo, which is man. Microbicide kills microorganisms. So notice that when we look at the total cell count, which is in red, you don't really see, I mean, you see them stop increasing, but you can't tell what happened, right? Because the total number of cells stays the same over time. So notice that would look the same. You have to actually grow them in order to see whether something is microbicidal instead of just microbistatic. Microbicidal, you have to try to put them in the incubator and grow them. And if now there are fewer cells over time, you know that you're killing them. So we have some things that are microbicidal, and some of those things are microbicidal because they pop cells. And we call those microlytic things. Microlytic things, like some antibiotics and like heat, for instance, right? We know that heat causes denaturing of enzymes and it causes thermolysis of the cell. So heat, right, would not only reduce the number of viable cells over time, but because they're lysing, you're not going to see them on a microscope slide because they're gone, and they're not going to be absorbing light in a spectrophotometer, and so you will see that decrease over time as well. So we classify things as static, cidal, or lytic agents based on what they do to control the microbial growth, right? So these are controlling by killing, right? So maybe that'd be heat, right? Or some other thing, and maybe this is cold, right? Remember, cold doesn't kill them, it just stops them from undergoing binary fission. Sometimes things have a particular way of operating, and sometimes it's a matter of concentration. Low concentrations will be static, and then maybe cytal. High concentrations will be cytal. But typically, you'll move from static to lytic, and that tends to be things that affect the cell wall, and then something in concentration can go from static to cytal. Yes?
Would like a lytic thing be the salt content because they like lyse, or will not do that because of like?
Well, so if you were talking about an organism that doesn't have a cell wall, so if the salt, or sorry, if there's a higher salt concentration inside the water rushes in, you could have lysis of the cell. But most bacteria have a cell wall that keeps them from lysing, but if you have something, right, because we're saying microbilytic, so lots of protozoans don't have cell walls, and so they could lyse. Alrighty, so a little bit of terminology. Now, before we talk about particular treatments, we want to think about things that affect all treatments, right? So what affects whether a given treatment will work? And the first thing is how many bacteria you're trying to kill. Makes sense, right? Think about how, like, laundry, right? It's the bane of my existence because it never ends. Like, every time I'm doing a load of laundry, I come back into the house and there's dirty clothes in the laundry basket, right? So when you see the massive load of laundry that you have in front of you, you know it's going to take you longer to do that laundry, right? If you only have a little bit of laundry, it's not going to take a lot of time. Same thing here. Lots of microorganisms to kill take a longer time to kill. And the really interesting thing about that is it's because it's protective, right? Just like that huge pile of laundry, right? You take the clothes from the outside of the pile and you put them in the washing machine. When you spray Cavicide on your desks, the Cavicide hits the bacteria on the outside and it kills them. But all the bacteria underneath are protected because the ones on the outside are protecting them. So what you have to do when you have a high microbial load is you have to give it longer, right? So a couple of you have had spills, and what I will say is spray the counter with Cavicide, leave it for 10 minutes. Because when you generally disinfect, right, when you sanitize your countertops, you spray them and you wipe it off because assumably there's a low microbial load there, not a lot of bacteria. When you take a test tube full of bacteria and dump it onto the countertop, are there more bacteria there now? There are, right? So we have to expose them for longer to the disinfectant because we need the disinfectant to penetrate that whole pile and get everybody. So it's not that underneath, right, are these highly resistant bugs, right, these little happy bacilli going, wah ha 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 ha, I don't care, right? If they are, their comeuppance is coming because the disinfectant is going to slowly make its way all the way down and the blammy, they're going to die too. But you have to give it adequate time, right? So the amount of time it takes to disinfect a given area or to sterilize a given area is dependent on how many microorganisms are there, right? And you can see that graphically here. Here's a low population load, right? Doesn't take very long at all for you to sterilize, right? Because now there's 0 cells, they're sterile. If there's tons of them, right, twice as concentrated a population, it takes twice as long to sterilize or to control the growth in any way, right? So the more contaminated something is, the longer we will let a disinfectant or another control method work, right? Like heat or any other thing that we would use. Now that is all microorganisms, right? So E. coli, staph, anybody, right? The more of you there are, the more protected some of the members of the population are. Sometimes though, the treatment will be affected by characteristics of the microorganism themselves, right? So for instance, if you have a glycocalyx, right? Remember glycocalyx, that's the very outside of the cell. It's this weird gooey polysaccharide layer, and it can be sort of thrown together like a, right, like a slime layer, or it can be highly organized as a capsule. So imagine spraying, right? So now you have over here a microorganism that has a capsule around him. I mean, look how big those capsules are. They're much larger than the cells themselves. And that capsule is going to absorb whatever's coming and keep that microorganism safer for longer. right? So a capsule. The mycolic acid in the cell wall of acid-fast bacteria, right, is wax. Today we're going to be doing acid-fast staining, and we're going to go back to that heat fixing step that we did with our smear and simple stain. And it says in your lab book, I think I even put it in bold, right, you heat fix, and when you set it down, you immediately flood it with the carbofuscin. And you have to do that, right, because it has to be hot in order for that carbofuscin to penetrate the mycolic acid layer. So generally, if microorganisms are lying on the countertop and you spray them, they're not hot, right? So that disinfectant is not going to penetrate them like it would if they were hot. So the mycolic acid layer. The lipopolysaccharide outer membrane that all gram-negative bacteria have is protective, right? It's basically just the more layers you have piled outside of the cell membrane, the more protected you are. So all bacteria typically have a cell wall, that's protected. The outer membrane of gram-negative bacteria is protective. The glycocalyx is protective. Anything that's outside is going to absorb and keep it from getting inside the cell. And all of these methods of control, they can't control until they get inside. Right? They have to get inside in order to affect the cell's growth. So some cells have structures that make them harder to kill, right? And of course, the one subcellular structure that we really think about when we think about that is the endospore. But there's a difference between the protocol for staining acid-fast cells and endospore-producing cells, right? The endospore, you're actually going to heat fix it 6 times. Heat fix it again, 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 and again, immediately flood it with malachite green. Because you have to get that spore, because it's so protective, really hot before you can get that inside there, right? So those are characteristics that not all cells have. Only some cells have those characteristics, but if they have them, it will make it harder to kill them, right? So that's why we think, right, when we think I want to sterilize something, I think, how do I kill the spores? Because if I kill the spore former, then I'm already going to have killed the mycolic acid producer, and I'm already going to have killed the one with the lipopolysaccharide outer membrane, and I'm already going to kill the one that's got the cell wall, right? Because if it took enough time to kill the spore, then it's killed all those other things prior to that, right? So that's characteristics of the organisms themselves. Another really interesting characteristic is moisture. So moist things sterilize faster than dry things, as evidenced by these data here. So this is a bit more sophisticated a graph because we're looking at 2 things, right? It's always kind of crazy when you have 2 y-axes, right? So we have here what we're kind of used to, the number of cells, right, over time. This one, if you want to track the number of cells over time, you need to look at the solid lines. So if we look at the solid lines, here is our wet culture, very quickly dies over time. Lyophilized means sort of freeze-dried. The freeze-dried culture stays the same over time, even though both of them are sitting in the same oven. Right? So one's in broth, and one's just freeze-dried, sitting on a piece of plastic. So you wonder, why is that? They're the same organism, same microbial load, right? Capsules in one, capsules in the other. Gram-negative for one, gram-negative. They're all the same. What's the difference? And the difference is one is wet. So you imagine you take this teeny little thermometer, and you put it in there, and what you see is that the temperature of this culture, which did not die over time, never increased. Because the dotted line is the temperature over time. So temperature over time never changes. So no doubt you're not going to kill them. If you don't change the temperature, you're not going to go up above their optimum, right? You're not going to cause proteins to denature, and you're not going to cause thermolysis of the cell. So the reason those cells don't die has nothing to do with them. It has to do with the fact that it's a dry culture. So when we sterilize things, we use moisture. Right? In the back room, we have an autoclave, and an autoclave uses heat that's moist in order to kill microorganisms, right? Because if we look at the wet culture, look at how quickly the temperature changes, right? The temperature quickly goes up, and that's because water conducts heat faster than air, right? And you can think about this, right? Do a thought experiment. Don't do this at home, right? But think about you're cooking a cake, right? Cakes usually bake about 350 degrees, right? You can put your hand in a 350 degree oven, right? If you're trying to figure out whether your cake is ready, you grab yourself a toothpick and you go right and you push it into the middle of the cake, and when it comes out clean, you know your cake is done, right? You're not gonna burn yourself. If you stuck your hand into a glass of water that was in your 350 degree oven, you would burn the crud out of yourself very, very quickly. Why? Because the water conducts the heat into your hand much faster than the air. Now, does that mean you can stick your arm in the oven forever? No, right? Ultimately, you will burn your arm, right? But it's going to happen much slower because water conducts heat faster, right? Same reason why you can freeze to death in 80-degree water. Because ultimately 80-degree water will pull your body temperature, right? Will pull the heat out of your body. It will be conducted into the water. All right, so moisture, right? Moisture allows that temperature to actually take effect, right? Because moisture is what causes the temperature to actually, change. The other thing, the last of these that we're going to talk about, is something that has several different names depending on what kind of microbiology you are studying. So the catch-all for that is organic matter, just stuff. If it's soil, it's dirt. If it's a wound, it might be dead cells or pus or something like that. If it's your piping or your ducting, it could just be stuff that's sitting there, right? Just muck. And again, that muck, all that muck does is block the treatment from getting to the cells, right? If the cells are sitting in a bunch of dirt, dirt's going to absorb the heat, dirt's going to absorb the disinfectant, and so they're going to be protected. So that's just stuff, right? Just stuff. There's a really special kind of stuff that also has more than one name, depending on who you talk to. In a medical community, these things are called vegetations. In the microbial sort of circles of the world, they're called biofilms. And these are not just muck. Biofilms are these crazy things that microorganisms can produce, and it's kind of like they make cocoons around themselves. If a cocoon was something where you could have your friends live with you and you'd sneak things back and forth and no one would know you were there, because look at what happens, right? So bacteria, they're cruising around, right? Cruising around your ducting, cruising around your urinary catheter, cruising around anywhere, and they're like, that looks like a nice place to set up shop, so they land there, because bacteria like to land on things, right? If you remember, we talked about the FTM, we put a little bit of agar there to give them somewhere to sit, right? Because they like to hang out. When they sit there, they start producing this chemical, and we don't care what it's called, so don't worry about it, right? But they produce this chemical that's kind of like bacterial cement, and that allows them to stick. And once they do that, they start to produce this biofilm architecture, and it really is like a crazy, like, have you ever seen tube worms? Like, you find a rock at the beach, and there's all these weird castings from worms that used to live. It's kind of like, they're these rigid structures. They're also, if you know anything about coral reefs, like a microscopic coral reef structure. So they can spread nutrients through the biofilm, they can remove waste through the biofilm, and it's incredibly resistant to penetration of heat, of chemicals, of anything. So when an organism can form a biofilm, it means it doesn't go anywhere. So like if they end up in the pipes of your sink or something like that, you actually have to roto-rooter them, right? You have to physically scrape them off of the pipes. If it happens in your ducting, you have to physically scrape it off of your ducting. If it happens in your urinary catheter, you have to remove that catheter because you can't get rid of it. If you are unfortunate to have one in your heart, you will have to remove that heart valve because you can't get rid of the biofilm. So this, right, so I said in clinical settings, right, they're called vegetation. So this is a heart, this is the mitral valve, right, so those of you that know anatomy or physiology. Here's the valve, right, here and here. Can't see the whole thing, of course, because this is two-dimensional. This gigantic mass is a bacterial biofilm that landed on the heart valve and grew into this structure, right? You can imagine, right? That heart beats all the time, and that valve opens and closes, and that vegetation doesn't go anywhere. Although, unfortunately, what will happen sometimes, especially with vegetation in the heart, is the heart will bump hard enough for a piece to break off, and then that piece goes somewhere else and it becomes a septic embolus, where it lodges in a blood vessel, and then from the blood, right, and then they just start growing there as well. And so you can get those in your brain, right? You get sent to the emboli in your brain, you get them in your, right? Embolus just means blockage, right? So you have a pulmonary embolus. But a septic embolus is a blockage because of microorganisms. So these biofilms, or vegetations as they're called in clinical settings, right, are a big, big problem because it's hypothesized that every organism has the ability to make maybe a little tiny bit, right? But a lot of pathogens are pathogenic because they can do that, right? So they'll land on native structures like heart valves. They're also really like to land on prosthetics. So if you have a prosthetic joint, or you have, like hardware, like screws or nuts or plates that have been put in, they like to land on that stuff too. And if they do that, you're in danger of having to have whatever that is taken out because they can't clean it off. And you think, well, but I mean, what if they're sensitive to antibiotics? Why can't I just take antibiotics, right? But we got to remember, this is not a penetrative structure, right? You can't get through it. And just in case you're like, whatever, Krista, here are some data to show you what happens when you have bacteria hiding in a biofilm. So these are two well-known, I mean, this one in particular, but tobramycin and ciprofloxacin, which goes by Cipro, those are two well-known antibiotics. So they set out to see what does a biofilm or a vegetation do for the organisms that produce it. So they did some studies, right? Of course, all studies that are done well have controls, right? So here is the tobramycin study. And as a control, they grew cells with no antibiotics. So they could see how those cells grow over time. You can recognize those because they all have these little open circles and triangles and squares, right? So as you would expect, when we don't have any antibiotic, they all grow over time. And then you say, okay, well then let's add an antibiotic. So that's our closed circles. So the colored in circles, the colored in squares, the colored in triangles. And in a lot of the cells, right, they die, which means they are sensitive to that antibiotic. So antibiotic, tobramycin, should be able to be administered to that patient for whatever that infection is. But here's the problem, right? Notice one of these three looks just like the control. And it's not the planktonic cells that are swimming around. It's not even cells capable of forming a biofilm that do that. It's the cells that are part of a biofilm. Notice the cells that are part of a biofilm grow as if there is no antibiotic present at all. And pretty much the same thing happens with everybody's favorite ciprofloxacin, right? Although people like Z-Pax better than Cipro, apparently, right? But look at this one, right? Again, the biofilm colony is growing almost equal to the control. Here, the same thing. Biofilm colony is growing almost equal to the control. So what that says is, it seems like there's no antibiotic there, but there is. So what does a biofilm do for you? It protects you from all the nasty stuff in the environment that would otherwise attack you and kill you, right? And that includes toxins, your immune system can't get to them, antibiotics can't get to them, so our choice is, right, to mechanically remove that structure from the body, wherever it might be. So, yes?
So if they don't move and they're just sitting there, what effect does it have on you?
Well, like if it's in your heart, I mean, this person's heart's not working, right? So you're not circulating, you can imagine, right? So what you'll do is you'll sound like a murmur, right? Because there's turbulent blood flow there. So there are a couple things that can happen. First of all, you're not going to get good blood flow. The other thing when you have turbulent blood flow in your heart is it causes your blood to clot. So you'll start throwing clots, and then you'll get pulmonary emboli, right, and other things as a result of that. Also, we're going to talk later about different things that bacteria can produce that make them more pathogenic, and a lot of that stuff like toxins can be secreted from this into the body. And they are sort of a reproductive structure, right? So they're here, but notice they'll release baby bacteria from that. So they're sort of like a little protective factory, and they just every once in a while shoot things out, and then they can go out to other parts of the body and cause more damage. And we haven't talked about like the pathogenicity of bacteria yet, but we will. All right, yes.
Is that a specific species that does that?
This, okay, my guess would be that's a streptococcal bacteria, and my guess would also be that that is a person who has some sort of valvular defect, right? So if you've ever known anyone who's had rheumatic fever as a result of streptococcal bacteria, those people will usually have to take prophylactic antibiotics anytime they have a dental procedure or any kind of minor medical procedure because their heart valve is damaged, which makes it far more likely that they'll get one of these, they call it bacterial endocarditis. But bacterial endocarditis can also happen as a secondary infection to a strep, like strep throat. It's one of the reasons strep throat is treated, even though it's a self-limiting throat infection, is those bacteria, if left unchecked, they make a beeline for even healthy heart valves and will attack them. And that you can die from that very easily. They call that, before the advent of antibiotics, green streptococcal endocarditis. Alrighty, let's see, so biofilm, yes.
So you can have like, if you treat strep throat, then like you can still have it there because the biofilm being foreign?
No, so you'll treat the strep throat. I mean, if you,
Like if it goes in
strep
there.
bacteria don't, so strep bacteria can attack the heart valve. They may not cause a big biofilm, but they'll attack the heart valve. But if you treat them with antibiotics and strep throat goes away, then you don't have that bacterium anymore. The idea with strep throat is to treat it before it becomes invasive. Yeah, so that's one of the reasons, and we're going to talk a lot about strep throat later on because it's one of our diagnostics that we talk about, but the idea is to treat what would otherwise be no big deal because it can go to the heart, but only if it gets bad.
Mhm.
All right, so, anybody else? I don't mind questions, you can always ask questions, it's fine. I may not know the answers, but, you know, I'll make something up and it'll sound good. So. All right, a little bit more terminology. So those of you, get your flashcards ready. Some terminology as, wait a second, did that go twice? There it is. Oh, I actually did twice. Accidentally did twice. So here's some terminology that talks about the control of microbial growth. The first is sort of a broadly used term, and it's called the decimal reduction time. So decimal reduction time is the time it takes to reduce cells to a tenth, right? So decimal reduction just means like if it's 100%, I take the decimal and I move it over 1. I've reduced the decimal by 1, and now I have 10 instead of 100, and 10 out of 100 is 1/10. So I have reduced the population to a tenth of what it was. So notice we have a graph here. This is the survival fraction, so the percentage of the population which is alive. So we start at 100%, we go to 10, and to 1, and to 10th, 0.1. Those are each decimal reductions, right? So from 100 to 10 is a decimal reduction, from 10 to 1 is a decimal reduction, and from 1 to 0.1 is a decimal reduction. So the question is, how long do I have to do a particular thing for that to happen, right? So what you're looking here specifically is heat as a control, but we can talk about the decimal reduction time for any control method we want to. We could do Cavicide, we could do bleach, we can do anything we want, right? This just happens to be heat because the other two are particularly about control of microbes using heat. So the decimal reduction time is the time it takes to reduce cells to a tenth, right? Because deci always means a tenth, right? Like people misuse the word decimate all the time. And you're like, oh, it decimated everybody. You're like, that means it killed a tenth of them. Which is probably not what you were expecting, right? When you said that, you meant like devastated, right? But when you say decimate, you mean remove by a tenth, right? Take a tenth away, not be left with a tenth, take a tenth away. It's fun, right? Fun word goes back to Roman soldiers and what do you call those guys? Deserting people, and they would just shoot 1 out of every 10 so that they'd still have an army, but there would be some sort of punishment for deserting the Roman army, right? Anyway, decimal reduction time, right? So this one, all the way down to a tenth of what it used to be. So the decimal reduction time notice for these, we can describe a decimal reduction time for all three treatments, 50 degrees, 60 degrees, 70 degrees, right? So the time it takes to reduce to a 1/10 at 70 degrees is maybe like 3 minutes. The time it takes to reduce the population to a 1/10 here, right, is about 11 minutes. The decimal reduction time for 50 degrees is 40 minutes, right? So if you don't have access to higher temperature, makes sense, right? You've got to expose them for longer, right? But that's a helpful term. It also can help us to sort of, you know when you say you can't compare apples and oranges? If you can come up with a decimal reduction time, you can compare ultraviolet light to X-rays to bleach to capacide and to anything you want, and you can rank them according to their decimal reduction time. So you can kind of take apples and oranges in terms of control of microbes and sort of standardize what you're saying. Because if you say X-rays have a decimal reduction time of 5 minutes and 50 degrees Celsius has a decimal reduction time of 10 minutes, you've got two things that are totally different, but you can say this one's better because the decimal reduction time is shorter. Like I said, the last two are particular to thermal, right, to heat. So we have the thermal death point and the thermal death time. Notice also not reducing, but killing. So these have to do with sterilizing, right? So death here means death of the whole culture, not death of a single cell. So how much time does it take to kill them? Is basically what both of these are trying to answer. One of them is you've locked in the amount of time that you have to spend, right? So the first one, thermal death point, says you have 10 minutes, and that's it. How hot do you have to make it to kill everybody within 10 minutes? So the point is 10 minutes. What's the temperature? So there's one thermal death point for every organism, right? Because that is the time that it takes to kill all the cells within 10 minutes. So if these are all the same organism, and one is exposed to 50 degrees, one is exposed to 60 degrees, and one is exposed to 70 degrees, if we want to know the thermal death point, we have to see when they get to the bottom. And this is where 70 degrees gets to the bottom, this is where 60 degrees gets to the bottom, and 50 degrees we don't know because we stopped the experiment before we could get the data. So the thermal death point says 10 minutes. So we go right here on the graph and we say, how hot does it have to be to kill them all in 10 minutes? And you say 70 degrees. 60 degrees is not hot enough, 50 degrees is not hot enough. Now we don't know if it's higher, right, or sorry, lower than 70. We know 60 won't cut it. And from our experimental data, all we can say is 70, right, is the lowest temperature where we will kill all cells in 10 minutes. So 70 degrees is the thermal death point. So the thermal death point is always expressed in just degrees. As opposed to the thermal death time, because here we'd have time as a variable and temperature as a variable. Time was a given up here, it's only 10 minutes. But now we're asking a different question. You can have all the time you want. How long does it take at a given temperature? You can have whatever temperature you want, you can have as much time as you want, just tell me how long it takes to sterilize it, right? So for that, we still can't get data for all three temperatures because what have we never managed to do at 50 degrees, according to these data?
Kill them all.
Kill them all. So we don't know anything about the death because they're not dead. So we have to go, At 50 degrees, all I can say is it's more than 50 minutes. That's all I know. At 60 degrees, though, how long did it take to sterilize the culture? 40 minutes. So the thermal death time at 60 degrees, notice we have to say that is 40 minutes. So there are two things that you have to give there. Whereas the thermal death point is just temperature, 70 degrees. Thermal death time, we have to say at 60 degrees, it's 40 minutes. What about 70 degrees? At 70 degrees, it is 10 minutes. So notice there's overlap there, right? Because that happens also to be the thermal death point, because it's 10 minutes. And that's okay, that can happen, right? So decimal reduction time is just any control. Thermal death point, thermal death time, those are particular to heat, which you could imagine because their names are thermal and thermal. Thermal death point, 10 minutes. Only 10 minutes, always only 10 minutes. Thermal death time, all the time in the world. Just tell me what you need to get it done. So those are terms that we're going to try to figure out in lab. We're actually going to be exposing bacteria to high temperature. We're going to have 3 different temperatures. Probably you guys will be 60 degrees, you'll be 80, you'll be 100. And we're going to take 40 minutes and see if we can kill them over that 40-minute period. And once we get all of our class data together, we're going to see if we can determine the thermal death point, because only one table will know the thermal death point. But we're also going to try to see if we can determine a thermal death time at 60 degrees, at 80 degrees, and at 100 degrees. And we're also going to do a fun thing where we have a spore former and a non-spore former so that we can see whether that spore increases survivability, right? Does the spore former have a longer thermal death time or a higher thermal death point? We're also going to throw some UV light in there too, just for funsies, and try to figure out how to kill microorganisms with those 2 methods of physical control, right? Because we're adjusting a physical part of the environment, right? UV light would be changing the light that they're exposed to. Temperature, obviously, right? Heat is changing the temperature they're exposed to. And we're also going to do some stuff with chemicals. We're going to do antibiotics and disinfectants and antiseptics and all sorts of fun stuff before we're done with all of our microbial control labs. All right, a little bit more on heat. We actually have 2 ways of sterilizing with heat. One you do, and one is done for you. The kind of sterilizing that you do with heat is called dry heat. And that is when you take your inoculating loop or inoculating needle and you put it in the flame of your Bunsen burner. And you do that until it gets red hot. Why? Because it takes lots of heat to sterilize when it's dry. We know that now, right? Because dry heat is not as penetrative as moist heat. So you spend a relatively long time cooking your needle and cooking your loop so that you can make sure it's sterile. So flaming, which is what you do, or you could do a hot air sterilization, which we never ever do unless we really, really have to, because it takes a long time, right? If I bump down here to the bottom, you wonder how long does it take, right? So a hot air sterilization, 170 degrees for 2 hours. I don't know about you, but I really don't want to sit here for 2 hours after we're done waiting to do something. So what if I just add moisture? If I add moisture in an autoclave, 121 degrees, so that's nice, doesn't have to get as hot, and look at this, 15 minutes. That's how much moisture changes that. So when you send your stuff to the morgue, right, they're all moist, because they're all in liquid, or at least when they get hot, the agar will melt and they'll be in liquid, and we put them in a sort of steam bath, which looks something like this, and we cook them. High temperature, high pressure, 15 minutes, everything blows, and they're done in 15 minutes, right? And that moist heat doesn't incinerate, right? Incinerate would be like your, if you did a bonfire, right? Take wood and you turn it to ash. And when you take what's on your loop and you turn it to ash. In fact, some of you may start noticing that you have new loops right now, so it's not common yet. But as you do more and more sterilization, that ash will build up on your loop. And when you inoculate broth, the ash will fall off into the broth, and you'll see it. You go, uh-oh, there's little tiny black things in my broth. But that's just sterile ash, because you have incinerated the bacteria that were on your loop. In the autoclave, we use heat to denature proteins, right? And that works just as well. Now, the incineration, of course, will lyse all the cells, we don't see them anymore. The autoclave will denature, and you think, oh, then there's still cells there. Well, it's hot enough that it actually causes thermolysis of the cells. So they die and then they lyse. And that's done in an autoclave. Autoclaves are meant to sterilize. Moist heat can also be used to pasteurize, but pasteurizing is like sanitizing. It doesn't sterilize things. So sanitization talks about inert surfaces. Pasteurization is kind of the same concept, but in food, right? And it was Louis Pasteur. I think the first thing he pasteurized was wine, because obviously that's the most important thing. And then they started pasteurizing other things. But it's not sterile, right? It just reduces the number of spoilage organisms, right? Because there were wine diseases. And then the French couldn't sell their wine, and that was a big deal. So he said, you should pasteurize it first, and then you can add whatever yeast you want to it, and then you get yummy wine, right? So that used to be a process that was kind of lengthy, right? 30 minutes at 63 degrees, which would cook whatever it was that you were pasteurizing and change the flavor and texture and different things about what you were working with. But today we have more modern pasteurization, and they're all equivalent, which is crazy, right? And the crazy thing is, look at the difference in the amount of time, right? So we have from 63 to 72 at 9 degrees Celsius, which granted, I mean, that's more than 9 Fahrenheit, right? But 9 degrees, you go from 30 minutes to 15 seconds, which is crazy. Not 30 minutes to 15 minutes, 30 minutes to 15 seconds, right? Now, you don't have to memorize any of this stuff, right? But that small increase in temperature has a drastic change on the microorganisms, right? And if you can crank it to a flash of 135 degrees Celsius, it only has to be for less than a second, which means that you can maintain the integrity of the food or the drink that you're pasteurizing, so it still tastes good, it looks good, right? It's not curdled, and you also have cleaned it, right? You've made it sanitary. You haven't made it sterile, so we still have to put it in the fridge or in the freezer, but it's safe for human consumption, right? So what happens when you can't heat something, right? What happens if whatever that thing that you need to use is what we call heat labile? It gets damaged, right? A really common thing that is heat labile is antibiotics. You can't sterilize antibiotics with heat, right? But you have a vested interest in having sterile antibiotics because you don't want someone injecting you with antibiotics that are contaminated, right? Because then you've just made a bad thing worse. So what do you do if you can't sterilize with heat? You can actually physically remove microorganisms, right? Which is hard because they're tiny. So if you use a filter, right? So we have these ways of filtering out microorganisms, physically removing them, right? Like if you ever make pasta, right? You have your noodles boiling in the water, right? And then when they're cooked, you pour them into a colander or a sieve, and the water rushes through the holes, and the pasta is stuck. So think about that, right? Little penne pasta, almost looks like a bacillus, right? Think about the broth, right? The broth goes through sterile, and the little bacteria are stuck. And so now what you have is called a sterile filtrate. So you can do that with antibiotics. We push antibiotics through these sterile filters, and then what we have left over is some sterile broth or filtrate that we can use. So I have some of these so you can kind of get an idea of what they are, right? So this, you buy them, they've been sterilized, right? This one's no longer sterile because I've had it in here for like 5 years, but this is where you put your dirty stuff. And you can even attach it to a vacuum so it'll create suction, and that white thing, that's the filter. So it drains through, and this is a sterile container that catches what gets through. So all the bacteria end up on the filter, and then all of your sterile filtrate ends up here. So I can make an antibiotic solution, right? I can buy a powder of antibiotics, and I can stir them up in water, and I can put them in here, and the antibiotics will be collected here in a sterile filtrate, and any bacteria will be collected up here. We also have these tiny little cuties, right? This actually has a filter inside it. So this would be in a sterile package, and you go, oh, I need to sterilize this fluid, and you pull it up, and then you aseptically put that on there, and then you use your manpower to push it through and collect it in a sterile receptacle, and now you have something that's sterile. So these are important because whatever it is that you're wanting to sterilize will get damaged in heat, right? It'll kill the cells, and it'll also kill your thing, which is not super helpful. And this is kind of a fun picture. That is a scanning electron micrograph of a filter. Here are the little holes that all the filtrate went through, and here are the bacteria stuck on the other side. You know, that's how they discovered viruses. They had what they thought was a sterile filtrate, and it still caused infection. And they were like, how did something get through there? Because that hole is so tiny, right? I mean, look at the size of that hole. That's 1.5 microns. What can get through that, right? We actually have much smaller holes now, and they still, they were like, there's something in there, and they just called it infectious filtrate. They're like, But today we don't do that because we know about viruses. So when we filter with things that will collect viruses and bacteria. So we have heat as a physical method of control. we have filtration as a physical method of control, and we can use light as a physical method of control. Which seems very strange, right? Because when you think light, you think stuff you can see, right? Which we think is a benefit because it allows us to see things, right? But when you talk about light, like what is emitted from the sun, a more accurate way of describing that is radiation. You're just able to see some of it as visible light, right? But that's a very small portion of what gets shot off of the sun all the time, right? If we look at all the things that get shot off of the sun, we have gamma rays, right? So you can make yourself an Incredible Hulk. We have X-rays, we have ultraviolet rays, we have this little skinny portion that's all the visible light, and then infrared radiation, microwaves, right? Not just coming from your microwave, but coming from the sun, and radio waves, right? We can also manufacture radio waves, but there are radio waves being emitted, right? So some of that radiation we like, because it allows us to see. But when you get to the lower end of the spectrum of light, you get, even though it's lower, it's higher in energy. So ultraviolet light is higher in energy than visible light. And X-ray radiation is higher in energy than ultraviolet radiation. And gamma rays are more energetic, right, stronger than X-rays. So gamma rays are so strong that we don't mess with them, right? We don't use those as a method of controlling microbial growth. But X-rays we can contain, right? I mean, you get X-rayed, and usually they'll put, like I just went to the dentist, because that's what you do every 6 months, right? And every year I get X-rays, and they put the big lead shield on me, right? And have a little thing that directs X-rays just here, right? And then I have something to protect me. So we can protect against X-rays, so we can use them as a method of control. And the way that they work as a method of control is by creating reactive oxygen species, right? Oxygen can become a superoxide, which can become hydrogen peroxide and all sorts of stuff. And superoxides, right? So in a cell, which is almost all water, you can shoot an X-ray and some of that water becomes ionized into these reactive oxygen species. And the reactive oxygen species damage DNA, damage fatty acids, damage amino acids, damage all that stuff, and the cell dies. Which is why when you get an X-ray, they cover most of you with lead plating, right? So that you don't have that stuff happening where you don't need the X-ray to go, right? So X-rays are strong enough that they can actually ionize radiation, water and cause free radicals. And that's how X-rays can be used to control microbial growth. It actually shines on the microorganisms, damages their DNA. If you don't have good DNA, you can't replicate your DNA and you die. Now, ultraviolet light is a little less energy, so it does not have the ability to ionize water, which is a fairly stable molecule. What ultraviolet light does is actually absorbed by DNA. DNA will absorb ultraviolet light, and it causes, right, in one of the DNA strands, right, in a double-stranded molecule, it will grab either two thymines or two pyrimidines, right? So, you know, nucleotides are either purines or pyrimidines. So pyrimidines can be hooked together when the UV light shines on them, and it makes this little hiccup in the DNA molecule. And you might think to yourself, well, I mean, what's the big deal about that? But when DNA polymerase sees this, it goes, oh, that's a T. I'm going to put an A across from it. And you just had what you learned in general biology is a deletion mutation, which is a frame shift mutation, which screws up everything downstream because DNA polymerase got confused. DNA polymerase is like, oh, this one, right? So imagine when you're out in the sunshine, right, you get a little sunburn. What have you done? You've caused all of your skin cells to absorb UV light. You've caused thymine dimers, and your body goes, I don't trust those cells anymore, and it sloughs them off, right? They die because you've damaged them with ultraviolet light. That's what happens when you get a sunburn, right? And the reason that every sunburn increases your risk of skin cancer because some of those cells got damaged not enough to die, but enough to have mutations, and if those mutations accumulate, you can get cancerous mutations, right? And that's why sunburn, right? So what does your body do to protect you? It makes brown. Because that brown, the melanin in your skin, will absorb the UV light and block it from the cells underneath and keep them healthy, right? So that's why people with darker skin are not as prone to skin cancer, because their cells are naturally protected by the melanin. And right, the underlying thing there is that UV light, right, is absorbed by DNA. It causes these dimers, right, dimer meaning two of them stuck together, which causes mutations in the DNA. So, because these are higher energy, even though they can be used, we don't use them in class, because we don't have a special room with lead and all that kind of fun stuff. So we're not going to do any labs with x-rays. We are going to do a lab where we try to kill cells with ultraviolet light, because ultraviolet light is really easy to block because it doesn't have that high energy. In fact, that plexiglass in the hood will block ultraviolet light. So what you see with the two little bottles, the things that you dried your gram stains on, that's an ultraviolet light. So we're going to use that in order to try to kill bacteria with ultraviolet light, right? Try to cause so many thymine dimers that we cause so many mutations that they kill the cells. All right, so that's it for the physical control methods. And I think we'll stop there, with the exception that I want to show you some lab stuff, right? So, just to make sure, hopefully everybody saw a plate that looked like that, right? I mean, I looked at your data. By the way, everybody loved your plates. They were super excited. They got looked at in all the other classes, and all the other classes are now going to do what you did because they were so impressed with you, right? The other thing you looked at was motility media, right? So we have this motile facultative anaerobe, right? Proteus is a facultative anaerobe, so he can grow with or without oxygen, so he grows through the whole tube. Pseudomonas is an aerobe. So Pseudomonas grew all on the top, and it can grow here because when you stabbed with a needle, you made a little hole that introduced oxygen. But he has a hard time growing around the whole thing. Then you had two organisms. I'm trying to think. Staph is a facultative anaerobe. I think Micrococcus is an aerobe, but I don't remember, right? But they were both non-motile. Everybody's red because this is a redox indicator, which we talked about last time, right? Now, you don't need to know this for lab, but we've talked about redox indicators, right? When things get reduced or oxidized, it changes color. So in the medium, it was clear until bacteria grew and caused that redox change, and everybody turned red, right? And two non-motile guys. I chose this one, because notice it's a little bit leaned over right there. That's because the needle got dragged a little bit. And the best way to tell whether you drag the needle or you actually have a motile organism is just to turn it. If you turn it and it changes shape, right? Like if you're looking at it from one direction and it looks like a nice line, and then you turn it 90 degrees and it kind of looks like that, that's not motility. You went in and across, and it's just growing where you put it. So if you ever have something that you feel like is ambiguous and you can't tell, because you are going to need to recognize motility media inoculations, right? You have a test coming up in lab too, and one of the things that you could be asked to do is look at motility media and decide, is that motile or non-motile? And if it feels tricky to you, just spin it around. If the shape stays the same as you spin it around, it's probably motile. If it changes, right, because if you see my hand like this or like this, it changes the shape of my hand, right? So that's non-motile. All right, one thing that we're going to be looking at today is spores. And this is a gram stain of spores. So I want you're going to gram stain your spore preparations as well as spore staining them today, because I want you to see what gram stains with spores look like. And some of you got to see that on your Ubiquities broths, but we're going to do that again today. And in the spore stain, spores are not green because it's not a spore stain. Spores are clear because you have not used a bunch of heat to try to penetrate them. And lastly, your Ubiquity data. You all have this. So I was a little bit surprised at the number of you who said inconclusive results, because I had 3 people tell me that they couldn't find things. You should not leave lab not having done it. Does that make sense? Like if you can't find them, you should reach out to me so I can help you. Because if you don't find things, you're not learning things, and if you don't learn them, then when you have a test, you may not do well in that portion of the test. So that doesn't mean that anybody's busted for this, but you don't want to go home without having told me, I can't find anything, right? Because I would like to help you find things. So you're going to have this in your lab notebook, right? So now I could ask you, what did student number so-and-so have, or what was this one, right? But our general thing is, are microbes ubiquitous, right? What does ubiquitous mean? Everywhere. So are they everywhere? Yes. There wasn't anywhere that there weren't microorganisms. So the big question of are microbes ubiquitous was answered yes by your class data. You individually had data about a particular place. And now you have data about lots of different places. Now not everywhere, but for what we've tested, we have not found a place where there are no microorganisms, so we would say that they're ubiquitous. Okay, let's take a break and let's start back up at 2:30.
So inconclusive